and privilege for me to welcome you all on the behalf of Arulmigu Kalasalingam College of Pharmacy for this 38th International Virtual Learning Series. First of all, I put forth my sincere thanks to our management for giving giving helping hands wholeheartedly to conduct this International Virtual Learning Series. Today, I am very delighted to welcome our resource person, honorable resource person, well-known researcher and undisputed researcher, Doctor. Can you hear, sir? Can you hear?
Yes, sir.
Yeah. Undisputed researcher, Dr. Scientist and Sunil Kumar, technical manager, US and Canada, DFE Pharma, USA. It is a great pleasure. It is a great pleasure for all of us. It is great pleasure for all of us that he could find time to be with us in spite of his busy schedule. Let me acquaint you with the sterling quality of this eminent personality, who is a well-known researcher. We are fully appreciate your for finding the time to be with us despite of your busy schedule, sir. Most welcome, sir.
Thank you.
Dr. Sunil, sir, most welcome you, sir.
Thank you very much.
Yeah, I hope this session will be a good platform for the students to get a good knowledge. So all the students kindly utilize this great opportunity and explore themselves. I trust that every students will put their effort to uplifting their successful career. All the very best for all the students. Once again, I welcome you all. Thank you. Thank you.
Thank you very much, Dr. Venkatesan. I mean, I'm humbled by the kind words, but yeah, I'm a learner. Still, I have a long way to go to catch up on all the recent developments that is happening in the pharma.
Thank you.
But thank you.
Please. You can start that. Please, sir, you can start.
Okay. So what I thought is I'll discuss on the recent developments, especially. Can everyone see my screen first of all?
Yes, sir. No issues.
Okay, good to hear. And I suggest if somebody is, if no one is talking, please mute their mic so that we don't have any distraction in that aspect. Much appreciated. If anybody has questions, please feel free to ask in the comment section. I think Mr. Anbaraj will coordinate that. Okay, so the title says the specification setting for excipients, right? So whenever you file a dossier, when I say dossier, it's the ECTD, Common Technical Dossier, that I'm referring to, a package that you put forward for every drug approval, right? How do you make it smart? So I'm going to focus on smart specification setting with the recent update, especially from the ICH Q12 and introduction or the concept of CASA, right? So knowledge aided structured assessment. Is my voice audible, loud and clear?
Yes, sir.
Okay, good. So first of all, what do you mean by specification, right? So specification, ICH Q6A says it's a test of references, list of test references, analytical procedures, and so on. The most important thing to note here is specifications are identified, proposed, and justified by the applicant, right? So market authorization holder or one who submits the application. And then it's approved by the regulatory agency, right? So one has to pay extreme caution and has to be prudent in proposing a because this is what affects your later post-approval communication with agency. Yeah, I do here's let's see. Okay. Okay, so that's about the specification. Let's consider a scenario where you need to do a change of specification, or you want to update specification post approval, right? So you have a product submitted already to the agency. It can be EMA, it can be FDA, or even the DCGI in
Sorry,
India. Yes.
sorry to
Nataraj?
disturb. We can't see your screen, sir.
Okay, maybe there's.
Yeah. Now okay.
You're able to see now?
Yeah. No issues.
Okay, thank you. Good. So let's consider a scenario where one has to update the specification of an existing product, right? So in which case you have an acceptance criteria that the market authorization holder or the applicant would have set during the filing of ECTDD, and then you default, right? So see, again, when I say specification, the standards can come from your DMF, drug master file, or the pharmacopoeia, can be USP, EP, or where you're operating. And there are mandatory tests and there are optional tests, right? But the important thing to note is not all see that way. So usually it is thought that all the tests in the pharmacopoeia are mandatory. It is not the case, right? So especially in the US, if you see, there are mandatory and then labeling requirements. I'll come to the labeling requirements, which can be particle size, density, and other things. Now that is where most controversies start, right? So you have particle size and density, which is given by a particular manufacturer. We can consider DFE, who offer a lot of excipients. Now, if one takes excipient from DFE, so it comes with, of course, pharmacopoeial compliance. Also, it has additional parameters such as particle size and density. Now, for example, if someone has to update, right, for whatever reason, there's a change in supplier, right? So it can be from competitor to DFE, and then it raises a red flag, because usually no two excipient suppliers provide the same method for the labeling requirements, that is particle size. So if one works on the edge at sieve, another would release on laser or even mechanical sieve. So it raises red flag, so you stop it, right? So this is important to consider. Anburaj, I don't know if you see any questions as of now, but I encourage all to keep thinking, and please feel free to ask questions in the comment section. We'll address that towards the end of the presentation.
Definitely, sir.
Okay. Okay, so coming to the specification of excipients itself. So, like I said, there's a minimum requirement which is given by the pharmacopoeia. It can be USP, EP, or even IP. And then one has to really evaluate whether, based on the specific application, they have identified a CMA or not. That is critical material attribute, right? So, I have seen there is requirement from customer usually focus on tightening the specification, right? Okay, maybe on the face of it, it appears all good, that okay, you keep on controlling the specification to make it tighter and tighter. But if you think about from a holistic understanding, I mean, why do you do it, right? So if you keep on tightening it, you don't have any leverage for formulation flexibility or even regulatory flexibility. So one has to really has to think through what is it actually that the specification is causing for the excipients, formulation and regulatory perspective from a how do you say post-approval change, because you want, okay, you have developed a product, you have filed the product, and you want the product to stay in the market for its shelf life, right? So you don't want FDA to pull the plug and abruptly because you have set the specification incorrectly, or it is not flexible in that case. So what is suggested is, in absence of CMA, so you evaluate all the parameters. So for example, a formulation needs some particle size requirements, for example, sustained release, HPMC particle size, or even the viscosity of HPMC may play a big role in your sustained release performance of the formulation. In that case, it makes sense. But for immediate release, usually the particle size, especially if you're working on uncomplicated product, I would say, okay, you not simply define uncomplicated product, but a simple formulation or otherwise, which is not a complex generics, you can try to work around with just complying to the pharmacopoeia, right? So if needed, of course, application-specific CMA has to be evaluated and made possible, but then you need to really evaluate. Otherwise, you'll be caught during the post-approval change. So looking into the ICH Q12 itself, what it aims, right? So this was formally combined and put forward in November of last year. I suggest all to, of course, go through the ICH website. It's quite interesting. They've also come up with ICH Q13, which is continuous manufacturing. I think I see that as way forward for the pharma industry, right? So there is someone coming. I'll allow him. Okay, Anburaj, I think there's Dr. Rajapandi. Okay. Okay, fine. So what it means is to get the best out of ICH Q8 to Q11, right? So there's already guideline for each of the pharmaceutical development, risk management, quality system, and also development manufacture of API. But then, like I said, it becomes silos, right? So how do you combine the best of all the guidelines, all these guidelines, and come out with the concept, especially the EC that I mentioned, established conditions. Anburaj, Venkatesan, sir, I hope everybody can still hear me, right?
Yes, sir.
Okay, good. Most important thing to note here is established conditions are legally binding. So you have to pay extreme caution when filing a ECTD, right? So, for example, you file a ECTD without mentioning what is an EC, what is not EC. In fact, Appendix 1 of the guideline clearly says, right, what of the CTD conforms or gives EC and not EC, right? So one has to really pay attention in that respect. So, sorry, it was quick. Okay, this is an extract of course, the Appendix 1, especially coming to the control of excipients. It clearly mentions specification per se is an EC, established condition, but there is, of course, right, so reading between the lines, justifications towards that is not an EC. So what it means is you filed a product with certain specification, and during the post-approval change, if you want to have an update, it is still possible, but it's very, very to and fro, back and forth communication with the agency. So instead of, so to make it more simpler and streamlined, you want to better describe, right? For example, I think all of you may be aware of the two-pack changes, scale up and post-approval change, which talks about change of a certain excipient and the levels of change, right? So 10%, 5%, and so on. That's the formulation. But then when it comes to regulatory, there are also levels of change, which also have come across. Again, I suggest to please read through the ICH and guidelines from the FDA. So the minimum change or the minor change is annual reportable. That means it does not affect your product that much, right? So also called the CB0, change being affected. 0, this is the level 1 change. Level 2 can be CB30, right? So any change that you intend to implement need to wait at least 30 days before you implement. If FDA has any query, they'll come back. If not, The most interesting part is if they don't have any, if they don't come back, it's a good news, right? So you can go ahead and manufacture with the change. And then, of course, there is the level 3 change, post-approval change, which you need to, of course, wait for FDA to give a green signal before you go to the go ahead with the updated change product or even the manufacturing. Okay. So, like I said, summarizing the best elements of Q8 to Q11, it talks about the EC in this guideline, established conditions, and then there is a reporting category, right? So the serial number 3 that is mentioned in the slide, that is what I mentioned about the formulation reporting category and also regulatory reporting category. So you need to really pay attention. So I think many of you will go on to join regulatory or even formulation or a QA department. You need to pay attention to usually, I mean, the practice is the checkbox style of working, which personally I don't like because they don't pay attention to what's coming in the future. So one has to be really prudent in seeing the form, the package as a living document, so and prepare for the future, right? So you need to really pay attention to that aspect. And of course, there's a minimum commitment needed, which is called the PSCMP, Post-Approval Change Management Protocol. That means what is needed from the agency is how well the market authorization holder or applicant is sure that the formulation will perform consistently over the year, right? So for that, you need multivariate oversight. I'll talk about the multivariate oversight in coming slides. But this is needed, and this is not new, right? So most of the big pharma have been already doing it for like 10 or 15 years. It is now, of course, the small players, especially the generics, to catch up. I mean, no offense to generic, many generics are also doing well, but also for large other generics who are not still participating to get into the multivariate oversight to make sure the product doesn't fail or even prevent certain failure that is seen. And of course, there are post-approval CMC commitments. What this means is not always the applicant has time to do all the test and parameters, so the applicant also commits to do certain tests post-approval. So after the phase 3, during the phase 4, those commitments can be met. Right? Okay, so the current way of working is what you see on the left, right? So ECTD, I'll of course not go into the details of the CTD triangle. Many of you might have been aware of this. The most interesting part is the EC that I talked about, and the CASA, which I'll come to that in a couple of minutes. So EC talks about, so how do you define, right? So is the process, in this example, is it a CPP, critical process parameter, or key process parameter? Yeah, it is in EC or not in EC. So it's not in EC, of course, please don't report it. So that's the essence of this presentation, right? As much as you pays attention to report in EC, you also pay attention if it is not, for example, I mentioned CMA, please don't mention it. Otherwise, you'll be caught later. That's about the EC. Now, coming to the CASA, or Knowledge Aided Structured Assessment, this is an interesting proposal by the FDA. This aims to reduce, how do you say, rather streamline the huge pile of data dump that we have usually in the ECTD, right? So there's so much unorganized data in here. So the knowledge aided structured assessment way of working aims to streamline it. Of course, I'll talk about the benefits of CASA in a couple of minutes. I talked about data dump, right? So if you look at the amount of data dump that we're experiencing recently, it's unphenomenal, or rather phenomenal, right? Unprecedented, it's phenomenal. So if you one study says there are about 62% of all data which is unstructured. That means you cannot make sense out of it. I mean, it's there. So what do you do with it? Especially you consider the 4 V's of data, right? There is huge volume, there is variety of things. I mean, what you do in social networking, and everything is connected back, right? So what you do in terms of networking, can be LinkedIn, can be slide share, can be your own academic data dump that is there, and there is veracity. That means there is uncertainty of the data. I mean, how sure are you that the data that was put forward like 5 years ago it's still valid, right? And there's velocity of data, there's continuous update of data, but how do you make sense of it? We'll talk about that. If you notice, now I started with specification setting and then the contributing factors, right? The contributing factors is the regulatory data dump and the formulation uncertainty, which I'll talk about in the next part of the presentation. But now we focus on the regulatory uncertainty and how to make sense out of the unstructured data. So EMA, European Medicinal Agency, has put forward thought process or a concept paper to effectively manage the data dump, right? So you have a lot of data around the quality, and then the formulation, the analytics, and other things. So they have put forward the roadmap for combining, or rather making sense of data linkage. So there's a concept of big data. I talk about also in slides. Excuse me. And then you make sense of it. So one way what is mentioned is you have a lot of unstructured data here in different silos, right? So different buckets, and then you create an entity-centric model. One of the way is multivariate analysis. Of course, I'll talk about that in the slide. So essentially, there's huge data dump. How do you make sense of it is you have a certain structure to it. How do you make sense from a holistic high overview rather than looking at the univariate data in that sense? Okay, talking about the case itself, right? So what it aims is to reduce the subjectivity. Now, if you think about it, you might have heard different products. So same formulation reviewed by different reviewers can give different results. So it is like same subject, almost written quite similarly by a candidate, has a different, different candidate get different marks. How is it possible, right? So that is part of that, of course, that is because of the subjectivity, right? So the CASA aims to reduce the subjectivity of the documentation and also improve the objectivity. Now, that's why I think objective-based questions are more rather practical, right? So there are yes is yes and no is no, right? So then there are 4 options and then you give an answer. So that is more objective-based testing, and that is what CASA aims at. Not only that, it also aims to use a lot of computer-aided assessments to, you work with the big data to extrapolate, extract, also understand the current data and also extrapolate for the future. So a lot of possibilities. It also helps to retrieve the historical data, which is currently, it is possible, but then it is very, very cumbersome. That's all about the regulatory uncertainty. Now let's look at the formulation uncertainty, right? Uncertainty. There is a concept called Q3. Now, for example, if I run a pharmaceutical company, Sunil and Company, for example, right? Now I file a product. Now that product can be, it has to perform what they call in general, right? So qualitative and quantitative sameness. But there's also, especially for complex generics, the structural similarity. Right? Just to explain simply, just consider this. So you take an excipient, you work with certain API. Let's say you work with the direct compressible platform, and you blend for a certain period, you document everything, you optimize, you also do the validation in the scale up and also in the exhibit batches. Now, what happens is when you then there's a change of scale from R&D to scale up, of course, the structural similarity, for example, blend uniformity, right? So usually blend uniformity or even the turret speed can affect your hardness and of course in turn your dissolution, right? Now the blend has a certain structure from R&D to scale up and post-approval, commercialization phase. Now one has to pay attention to that and has to study. I mean, typical excuse given by generics is, oh, they don't have time. But of course, if you don't invest in time today, then yeah, please prepare for having repercussions later in terms of product failure. So one has to really invest time in understanding the formulation to the fullest before it's submitted to the agency. So that's about the concept of Q3. How is specification given, right? For example, if one procures, I think many of you would have procured excipient from various companies, so a certificate of analysis is given. Now, does it always reflect the entirety or totality of the excipient, or it can be API or even the process? It's not, right? So it mentions, the certificate of analysis mentions certain parameters to say, okay, this excipient meets certain standards, but that does not mean everything about the excipient is mentioned in that COA, for example. Now, when you consider or compare with the nut and bolt, right? So the nut and bolt has all the engineering tolerances mentioned and captured. There is no two ways to it. Yeah. For example, typical excipient COAs would have the pharmacopoeial compliance, the labeling requirements, that is the particle size density, but rarely, of course, if it is application specific, then you would have the molecular weight, right? Or even the chemical composition. Now, lactose, yeah, so lactose, of course, has two polymorphs, lactose monohydrate and anhydrous lactose, but what about the impurities that it has? So you need to really investigate, and those things play a role in your formulation, right? So certain APIs may interact with certain excipients. It can be lactose, it can be MCC, it can be dicalcium phosphate and other things. And sometimes the impurity itself contributes to functionality, right? In terms of dicalcium phosphate, if you consider what is considered as impurity actually contributes to functionality, right? So without that impurity, dicalcium phosphate would not perform. So you need to really pay attention to that. Okay, so when you develop a product, right? So you of course do your DOE, you develop your QPD principle, obtain what are CMAs, what are the CPPs, and contributing factors towards the quality target profile, QTPP. But then what happens after manufacturing? So you need to also think about that, right? So it doesn't mean that if a product is performed for, let's say, 5 years, it will perform for next 5 years. There are many examples where specification has been same, the product has been performing for over how many years, maybe example of 5 years, and then for 700 batches, but the 701st batch failed. How can that happen? So that can happen because of what is called as unspecified attributes. Now, what you see, you can control, right? So you identify certain parameters and control it by ways of design space and other things. But there are several degrees of freedom, and that contributes from what I talked about in the previous slide, right? Not all parameters are controlled. So those parameters you can get out by ways of multivariate analysis. So those are called unspecified attributes. So typical, let's talk about the typical practice, right? So you start from the R&D, right? So you establish design space, you validate the or verify design space by edge of specification in the scale up and the manufacturing. Yeah, you don't pay attention most likely, but of course there are companies who pay attention. So you need to really monitor the unspecified attributes, where you monitor the known and the unknown factors affecting your formulation, right? So I'll talk about taking an example in the coming slides. How are we doing with time, Anbaraj? I think we're good.
Yes, sir. Yes, sir.
Okay. So of course there are swans there. If you're wondering what what does it got to do, see in Europe there was a belief that okay, so everything was white, right? So when you formulate, everything is rosy, right? Okay, you've identified your parameters and everything. So known parameters are identified and controlled. What about the unknown, right? So I'll talk about that. So in that case, like I said, yeah, everything is pleasant. But then one day, the 701st batch is black swan. That means there is OOS or out of specification. How can that happen? Is because the unspecified attributes are not considered, right? So now coming to the excipient version itself, if you look at this picture on the right, of course there are three manufacturers mentioned. Of course now it is two because was acquired. DME Fonterra and Bocolo Domo is now one company, and then Kerry is another company. Within the same manufacturer, within the there is also variability, and of course between the manufacturers there is variability, right? So variability is true. One has to really pay attention to understand what is the variability. I'll talk about how DFE does investigate variability and conform to product sameness. I also talked about the use of big data, right? So there is a paper from Thurens. Please feel free to refer that. What he has done is he has investigated product performance from two different sites, and then he selected few batches to represent the entire spread of the data. Of course, you cannot test the entire batch of like 120 or even 140 batches, right? So you just select the edge or batches to represent the spread of the data, right? So usually that is a 95% confidence interval. I'll talk about that in the coming slide. And even the multivariate way of formulating products is not new. So if you look at this picture, Novartis has already developed this technique way back in 2006 for a wet granulation product. So you see after dispensing and granulation and all the in-process unit operations, blending and tablet compression, right? So it continuously is monitored by the multivariate model, right? So any change, for example, in the particle size or even density of a certain excipient, then it raises an alarm, right? It's already programmed in the algorithm. Okay, now let's take example of continuously manufactured anhydrous product from DFE. So DFE comes out what is called the product consistency document, right? So this is a proactive thing, an agency, regulatory agency, and even the customers love it because instead of customer doing all the excipient variability, we help them with making sure that the product comes from the center spec, right? So if you consider two excipient manufacturers, the middle picture, right? Both conform to, let's say, between brackets, same specification, but you look at the noise that one is operating here versus here, right? So we aim, I mean, DFE aims to operate in center spec. And how do we make sure that product that customer receives is in the center spec is by way of multivariate analysis, right? So for example, we investigate, now we don't talk about the age of specification, which is usually talked about in the industry. We talk about complying to the center spec. I mean, narrower, tighter, but I would not say keep on going tighter. It doesn't make sense. And then making sure that it is within that space for years to come, right? So whatever we are going to see in the coming slides are the batches from the center spec. Just remember, I'm not talking about the age of specification, right? I'm talking about the batches from the center spec. A small introduction of the big data itself. Maybe you have come across this, but in the larger interest. So you have a lot of big data, unstructured data. How do you make sense of it is by simply put, right? So you have in the univariate data, you have the X and Y axis. In the big data or multivariate analysis, you have different what they call principal components, right? So you treat as X, Y, Z, or even you can have fourth dimension to it. So you typically end up with what is called the scatterplot or score plot, which shows how, with respect to a certain variable, the data is behaving. And the loading plot actually talks about what are the vectors, what are the factors that drive or cause this drift. Now, remember, I'm within DFE, we investigate the drift from the center spec, right? So that is here, not age of specification. That's important to note. So again, talking about the 21AN, super type 21AN is the anhydrous lactose, right? From the center spec. So this is a 95% confidence interval. This is a score plot. So score plot can be a cluster or many clusters, right? So for example, if there is manufacturing from different process trains or different manufacturing facilities, you may get two different clusters, right? Now, of course, the dots that you see here are different batches, now manufactured from 2014 to 2018. Yeah, I don't know if you see a pattern, right? So typically, batches manufactured from a year has to have in a single cluster. But if you pay attention and notice 2016 batches, right? So there are two clusters, one on the left and the right. And as a user, one doesn't have visibility to this, but with DFE, we always provide this because it is important. The agency wants to know, and customers, of course, wants to know this. So this was because of planned maintenance. Now, again, does this planned maintenance affect your product? Probably not, because we investigate also the applicability or the application of this, right? Now, this is a loading plot. So what you see here, for example, the blue batch versus the red batch, they're apart because of these parameters, right? So mainly the beta content of the anhydrous lactose, which is the most, which is found to be most driving factor, vector in this case to drive the differences, scatter, the color and ash. I'll not talk about color and ash. It is important, but most important is the beta content, which contributes to the functionality. Now, similar to the Thurin's paper, so we identified few batches to represent the entire spread of the data, right? So, of course, batches are outside. That means it is within the center spec, but then outside the 95% confidence interval for this particular data set. So we formulated a tablet with tremulose and magnesium stearate as excipient. So what we found was for a given beta content varying from 78 to 86%, so the tablet tensile strength varied from 1.2 to 1.75, I would say. So that means the neat excipient has this particular tablet tensile strength. Now we can also, this is the starting point, right? So now customers or formulators, what they do is they take this data and input into their formulation multivariate analysis. Just picture this, right? So similar to the excipient multivariate analysis, you can do also formulation. So for example, you can understand similar to this, what is the bulk density of this product doing to my dissolution, right? Or you can also include the process parameters such as current speed to affect your, to understand your dissolution per se. So a lot of possibilities, and normally in univariate understanding, you will never get this interaction effect, right? So I mean, only when you do a multivariate analysis, you will understand what is the interaction effect of product, I mean, different formulation variables and also the process variables, and also predict probably what's coming, most likely the failure, right? Like I talked about also the unspecified attributes. So known is controlled, unknown can also be um, studied in the multivariate analysis. This is the VIP, which is the variable importance for projection, also from the same software. What we understood was it confirmed that the most important parameter for this particular excipient is the beta content, followed by, of course, the relative alpha, particle size, and moisture in that respect. So I talked about the scatter plot, right? So you see, in this case, the Thurin's paper, you have two different clusters. Now, not always, it's not like two different plants or you get different results, right? So because there seems to be similarity within the product, in this case, of course, was MCC, in your finished product, which is a tablet on the right? So you see that is gone. So what you see as cluster for particular set of experiment, you may not see with a particular formulation example, in this case, tablet manufactured with the MCC, but it can also show clustering. And then you need to pay attention why to show clustering, is it important, and so on, right? So it is like you have a lot of data, but how do you make sense of it? So you need to continuously invest in understanding what do you derive out of it. Okay, after all that, so to summarize, we talked about EC, we talked about the regulatory uncertainty, we talked about formulation uncertainty. So how do you set really the smart specification, right? So suggestion is, of course, please investigate the CMA, right? So with respect to your formulation, at the minimum, of course, you comply to the pharmacopoeia. Of course, there are labeling requirements. These are formulation specific. If you have identified a CMA or certain CMAs, have them identified, controlled, monitored, and controlled. But if there are no additional CMAs, please don't include it, right? So it is not a CA. If you don't mention it and then file a dossier, the agency thinks that it is an EC. So we don't want to be in that situation later. So CMA, yes, if there are no additional CMAs, please don't mention it, or rather say that this is not a CMA or it's not an EC, right? So have always a multivariate oversight. So you can, of course, have a univariate approach. Okay, what does my hardness do to my dissolution and so on? But you should also understand what does your hardness of the tablet do to your dissolution or the turret speed or even the API composition or your excipient composition. It's important to know that. Okay, so how do you implement this, right? So a way of implementing is what I suggest, especially when I talk to customers, is please have your own method, right? So of course you can try to learn from pharmacopoeia and the customer's methods, but it also always saves the day for the customer or the market authorization holder if they have their own method. Then they have the flexibility to work within that method rather than relying on a particular vendor, and the vendor says we have changed. Now that raises a lot of red flags. You don't want to have that. Have multivariate oversight. Please review your post-approval, how do you say, product which is in the commercial manufacturing. So please review the performance and also look for any drifts, right? So if there are drifts, pay attention. It may be a warning sign. Of course, if you don't pay attention, there could be failure. And we have seen it, right? There are many examples for failure. Look for drifts, that's that part. And the beauty of multivariate analysis is you can know the drifts of the known attributes, factors, and you can also get an understanding of the unknown or the unspecified attributes. Good. So that's about a smart specification setting for excipients. You can elaborate on this to an API or process and other things. But yeah, the floor is open for questions, and Raj. So let's take some questions.
Thank you, sir.
Thank you, sir. Thank you, sir.
I'll stop sharing now. Let's take questions.
Sorry, ho.
Sunil Kumar, sir. We had a wonderful and informative presentation, sir.
Welcome, sir.
Yes, sir. On behalf of our management, I hereby express my wholehearted thanks to you and your management for giving such a wonderful and informative presentation, elaborated presentation.
My pleasure, sir. Of course, if
Thank
there are any
you. Thanks.
questions now,
Yeah.
of course, Anbraj and Venkatesh, sir, you have my email ID. Feel
Yeah,
free
yeah.
to connect with me for any questions. You have my presentation.
Yes, sir.
Please feel free to share it with the group so they may have the questions later.
So there are some questions, sir.
Yeah.
Okay, please.
Sir, actually, that material you have presented is very new to us as
Okay.
academic people.
Uh-huh.
There are a lot of new terminologies.
Mm-hmm.
Okay, sir. So, can you elaborate what is the difference between that structured and unstructured analysis, sir?
Oh, yes, sure. So, see, the current way of working is so there is a pharmaceutics division within the AKCP, right? So,
Yeah.
yes. So when you formulate something, what do you do? So you formulate, let's say, a tablet, for example, immediate release tablet. So you start with sourcing the API, sourcing the excipient, then studying which way to go for it, right? So you want to do the wet granulation or direct compression or even dry granulation, and then you document everything in your what they call product development dossier, right, in the
Yeah.
lab.
Yeah.
Now, in the manufacturing on the commercial setup, you do that via CTD, right? So, or the ECTD, Common Technical Dossier.
Those here.
Now, the Common Technical Dossier has elements of everything, right? So, when you do formulation, it's the 3.2 P.2, I think, which talks about so what are the parameters that you've studied and how do you control it? Now, this is from a univariate approach, right? So, that means, for example, for let's take example of a disintegrant, right? So if you worked with a primulose, cross-cumulose sodium, what happens if you work with 2% primulose versus 4% versus 10%? What does it do? What it does to your formulation, right? But then what is the impact of, since this is univariate, so X and Y, but what is the impact of this over your dissolution, over how do you say, the compaction, right? So that can be done, understood only when you do multivariate analysis. So you can, of course, there are control charts for studying the individual unit operations, and then you can combine everything to see what is the impact of, let's say, concentration of disintegrant over your dissolution, or even how do you say, the turret speed influencing, or the compaction pressure affecting your formulation. Did I come to the point, or?
Yes, sir. Yes, sir. And you have told about the CASA, sir, the knowledge aided.
Yes.
Sir, this is a FDA initiative or it's a part of ICH?
No, this is FDA initiative. So based on, I see that as a, how do you say, a continuum, right? So we have the ICH Q12, excuse me, which is
Okay,
just
sir.
concluded November last year.
Okay.
And then I see CASA to be the next step. So where the agency is going forward is, you have all the, I mean, basically, if you think about it, you have the Q8 to Q11, which is already there for many, many years. Now, Q12 is put forward to extract the best practices of all that. And then the next after that would be more of CASA, right? And so what
Okay.
it means for all the customers and even for the academia is, you need to prepare yourself to be smart, right? So you can
Yes, sir.
have old guidelines, but then what is it doing to you? So I mean, now it's like at the push of a button, you get everything, right? So it is like that. You can extract, you can do play around it, and so on. So yeah, it's better
Okay, sir.
data management, I would say.
Okay, sir. Okay, sir. Where we will get the data, sir? You have told about a lot of big data. So from where we are going to get the data, from FDA or from any other organization?
No, so it is the big data is coming from your formulation, Nataraj. Basically, say you investigate, for example, you filed a product and it is in the market for 6 or 7 years. So
Yeah.
you have the dossier ECTD, and you have all the when you have the minimum commitment, right? So after you file, you have to submit at least one stability batch data to the FDA every year. So you have that, right? So you have
Yes, sir.
all this data, and what you do with all this data is just by a simple data analysis software. I mean, in this case, what we have used is from Umetrics, right? It's called SIMCA. So you just understand the structure of the data. So see, beauty of that is, of course, it can blow up everything, right? What you see,
Yes, sir.
if you had paid attention to the color and ash, it is like very low value, right? But then
Yeah.
that is important because for an excipient such as lactose, color is the stability indicating parameter.
Okay, sir. Okay.
Yeah.
Sir, that CASA is used only for generic drugs or for
Anything. So
new
again,
molecules?
it is for anything, right? So of course, to implement CASA, it is not yet effective, right? So this is a concept from FDA. But of course, I'm sure it will come in the coming years, but we can prepare for it, right? So this is the preparation time that we have, but it can be applied to all, basically.
Okay, sir. Okay, sir. Thank you, sir. So one more question regarding that regulatory department. So some of the students they want to pursue career in the regulatory.
Mhm.
So what are the areas they should have sound knowledge?
I think one has to pay attention to all the recent developments I mean, across main. I mean, basically, ICH, as you know, is a tripartite FTA. I mean, US, Europe, and Japan. So one has to pay attention to uh, the regulatory developments in these three regions, and of course, I think the rest of the world mainly take it from there, right? So, of course, if you consider India, I mean, India, there's I think the second largest or the largest FDA approved facilities outside the US. So mainly we follow the USP and US FDA, for example, right? So I would say please follow ICH, FDA regulatory guidelines, and also the EMA guidelines. Japan MHW is there, but of course, if you follow, in my understanding opinion, if you follow the EMA and FDA, I think it should suffice for the Japan. Though there are additional requirements for Japan, but that's a different topic another day.
Okay, sir. Thank you, sir. If any other question is there, we will forward to you, sir.
Sounds good, but I really appreciate all of you taking time. Vegetation sir and Nataraj, thanks for inviting me, and
Thank you, sir.
I hope
Thank you, sir.
I have contributed something. But
Sir,
let's see.
in future also, let's have a good collaboration, sir, for the
For sure,
development of carriers.
for sure.
Kindly refer any of your friends or colleagues, sir, for our future webinars, sir. Please, sir.
Will
Kindly
do,
support
will do.
us. Kindly support us. Kindly support.
Always
Thank you.
do.
Thank you. Thank you. Thank you. Take care, sir. Take care.
Thank you very much, and yeah, you have a good evening.
Yeah.
Okay,
Thank
sir.
you.
Thank
Thank
you,
you very
and
much.
Take care. Take care.
bye.
Thank you, sir.
Thank you.